A valuable data type you will see as a clinical data scientist is laboratory measurements. Laboratory tests, also referred to as labs, are used to measure specific features of a patient's body and tissue. These tests help providers diagnose disease, plan and manage treatments, and track patients' overall health. Labs are some of the most objective data available in electronic medical records, making them very valuable for analytics. However, the wide variety of lab tests used in medicine can make it difficult to understand and apply these data. In general, lab tests can be broken down into two broad categories: anatomic and clinical pathology. Anatomic pathology examines organs and tissues, ranging from simply describing the appearance of these tissues by the naked eye or under a microscope, to testing biochemical and molecular properties of these tissues. Clinical pathology, often referred to as laboratory medicine, handles testing of bodily fluids like blood and urine. There is a broad range of tests conducted on these fluids. Clinical chemistry measures the presence of different chemicals and proteins. Microbiology identifies the presence of infectious agents. And hematology examines and counts the types of cells present in blood. Molecular diagnostics, or DNA tests, can fall under anatomic or clinical pathology, depending on whether they are using blood or testing a particular tissue, like a tumor biopsy. As you can imagine, lots of different types of results can come from these tests. Generally, anatomic pathology and molecular diagnostic results are provided as reports that are unstructured or semi-structured. Clinical pathology results, on the other hand, are often numbers or have limited choices like present or absent, and are usually stored in structured formats. Thinking through the 4 W's, laboratory data actually have a number of people involved at multiple time points interacting and generating data for a variety of purposes. Because of this complexity, we are actually going to break down laboratory data into different steps and address the 4 W's at each step individually. The first step in generating laboratory data is actually ordering the laboratory test. Lab tests are ordered by a healthcare provider when they need more information to assess or treat their patient. Put another way, laboratory testing only happens if the provider has a specific question in mind based on the patient's symptoms. The only exception are screening tests, which are run based on the patient's overall risk for a disease, even though they don't yet have symptoms. Screening tests are a public health effort to increase early detection of disease. So, what does a lab order actually look like? When ordering tests performed by clinical pathology, the provider typically requests the specific test they are interested in. For example, the level of sodium or potassium in the blood. Providers may also order a panel or a group of tests that are used together to assess a patient's health. Individual tests may be present on multiple panels. For example, sodium and potassium levels are tested in both basic and complex metabolic panels. Orders for anatomic pathology are typically less detailed, simply outlining the general clinical concern or diagnostic question and leaving the pathologist to determine the optimal testing approach after examining the tissue. The second step of the process is the patient visiting the lab to give a sample for testing. At this step, patients will encounter a lab technician who processes the lab order to identify what kind of sample is needed and how the sample should be collected. The kind of sample is called the specimen type and can be things like blood, urine, or even a stool sample. How the sample is collected is the method of collection. For example, is it blood drawn from an arm or a finger stick? Is the urine sample a random collection or all urine passed in a 24-hour period? The third step of the process is analyzing the sample. Laboratory samples are processed by laboratory staff and pathologists. Pathologists are physicians with specific training in conducting anatomical or clinical pathology assessments. For simple tests that have largely been automated, laboratory staff can process the sample at the same place it was collected and return the results very quickly. This is called in-house testing. Depending on the facility or the complexity of the test, many labs are sent to a different location for analysis. These are called send-out labs and can take a much longer time to receive results. Every lab, whether in-house or send-out, have their own protocols and processes for analyzing samples. These protocols define how the sample should have been collected and processed in step 2. For example, blood tests often require different additives to be added during collection so that the sample will be ready for processing. If you have gotten a blood test before, you may have noticed that the tubes were different sizes and had different color lids. These indicate what kinds of additives are in the tube. If multiple tests are ordered and the tests require different additives, patients will have multiple tubes of blood drawn. Importantly, for a number of medical tests available, there are actually multiple ways to measure the desired chemical or protein. Which technique is used can be different between labs or could even change over time within the same lab. All of the data elements described in steps 2 and 3, the specimen type, method of collection, testing protocol and results, as well as sample tracking information, are stored in a Laboratory Information Management System, or LIMS. The fourth and final step in the process is returning the test result from the LIMS to the medical record. However, only a few data elements are actually returned to the provider who ordered the test. The specimen and collection types may be returned as individual data elements, or they may be combined with the lab test name. For example, sodium 24-hour urine, or sodium serum. In addition to the lab result, for clinical pathology tests, a reference range will typically be provided. Reference ranges are the expected results if the test is considered normal. This can be an actual numeric range or something like not detected when performing a drug screen. Finally, multiple dates may be returned. The date the test was ordered, the date the specimen was collected, and the date when the results were available.